All right, I'm going to talk about cell organelles, an example of how they work together, and also cell size. So let's start with ribosomes. Remember ribosomes, they're not membrane bound, but we do sometimes call them an organelle. They make protein. They make it based on the order of RNA in an mRNA strand. In eukaryotes, you can find ribosomes attached to the rough ER or free-floating in the cytoplasm. In prokaryotes, they don't have an ER, so they're pretty much all free-floating in the cytoplasm of the prokaryote. Endoplasmic reticulum. This is membrane-bound, meaning we're only going to find it in a eukaryote, not in a prokaryote. The rough endoplasmic reticulum allows for compartments for some reactions to happen, and it makes some of the proteins in the cell. So some proteins are made on the rough ER, some proteins are made by ribosomes that are free-floating. Smooth ER makes lipids and detoxifies toxins that enter the cell. The Golgi body, or Golgi complex, or Golgi apparatus, it has many names, but Golgi is always there, folds proteins, modifies proteins, for example, adding a glycol group, so you can make a glycoprotein here, and packages and ships proteins out. So in my picture, this is the Golgi body. It comes after the rough endoplasmic reticulum. Next up, the nucleus. I think we all remember the nucleus is where DNA is stored. I drew it in orange in the center here. You'll see that it goes nucleus, rough endoplasmic reticulum, Golgi body. Eukaryotes have nuclei, prokaryotes do not. Prokaryotes just have their DNA free-floating. The mitochondria is where ATP is made from glucose. We'll go more into detail in that when we talk about cellular respiration in a different video. There are multiple, up to 100 mitochondria in a single cell. They're only in eukaryotes, they're not in prokaryotes. Prokaryotes have most of the proteins that are in mitochondria, so they can make energy too, but they don't have the actual membrane structure of a mitochondria. Lysosomes are membrane sacs that have hydrolytic enzymes. So let's remember hydrolytic, water, break. Oh, so these are enzymes that are breaking things down. So there would be multiple lysosomes in a given eukaryotic cell, and they would have enzymes for digesting either food that's taken into the cell or proteins or things in the cell that need to be broken down so that the individual amino acids can be used again. Vacuoles are membrane sacs that store things. So again, all of these that are made of a membrane are only in eukaryotic cells, not prokaryotic. Animal cells would have littler ones for storage. Plants sometimes have a central vacuole where they keep water as a way to make sure that they have enough water, but they're not, and they're maintaining their shape and they're not like shriveling up. So sometimes plants have a larger central vacuole. And plants can also have chloroplasts, and this is where glucose is made from carbon dioxide and energy from the sun. A quick reminder of the shape. There's thylakoids, these little membrane pancakes in them, and the space in between the thylakoids is called the stroma, and it's a double membrane. Again, we'll come back to this when we do photosynthesis. So let's look at how all of these structures relate to each other in terms of protein synthesis. So proteins in our cells either are going to exist in the cytoplasm, or they're going to in some way be associated with a membrane. So for example, they could be a membrane protein, they could be released through the membrane, they could be in an organelle, okay? And there's two pathways to make membrane-related proteins and non-membrane related proteins. The first is the simplest. DNA to RNA, they both start that way. The mRNA leaves the nucleus and then goes to a ribosome. If it's going to be a protein that's in the cytoplasm, it's a free-floating ribosome. Protein's made right into the cytoplasm. Maybe something called a chaperone helps it fold, but that's pretty much it, and then it just is there to do its job. So my favorite example of this one is like a motor protein that's going to walk on actin or myosin. Also, actin and myosin are examples of cytoplasmic proteins as well. If this is going to be a protein that is somehow associated with a membrane, it's going to go through the endomembrane system. This is the rough endoplasmic reticulum and the Golgi body. So this mRNA would go to a ribosome that would take it to the rough endoplasmic reticulum. The protein would be made into the rough endoplasmic reticulum, maybe into the membrane or into the ER itself. And then it's going to go through the rough ER, come out the other side, and be sent in a little vesicle to the Golgi body. In the Golgi, it might fold in a different shape. It might have a glyco group added, depending on what the protein needs to do. Then it's going to leave through the other side of the Golgi and go to its destination. So that could be to the membrane to either be released outside as maybe a signal, or it could be in the membrane and it could be a membrane protein ready to receive a signal. Or possibly this vesicle is going to take this protein to an organelle, like the mitochondria, or another organelle that needs that protein to function. Or this vesicle could even turn into a lysosome if these proteins are hydrolytic enzymes. And then it's going to just be able to be a lysosome and break things down that need to be broken down. Quick reminder about cell size. Why are cells so small? We did a lab in class where we had these agar cubes of different sizes, and we timed how long it took for their color to disappear when we put them into an acidic solution. Surface area to volume ratio matters. Smaller cells have more surface area per volume. So that's what we saw in the lab. We saw that the smaller cell finished diffusing faster, meaning all of the supplies that are needed for that cell can get into the cell faster than if there's a larger distance from the surface to the center. So we're looking to maximize surface area to volume. So for example, if we had a big chunk of a cell versus many cells in that area, it's going to take a really long time for the supplies that this cell needs to get to the inside. Whereas if it's lots of little cells, all of this surface area can have diffusion of oxygen or whatever else the cell needs happening all at the same time, and those cells are going to get what they need a lot faster than if it were a bigger cell. Some cells have adaptations to increase their surface area. For example, cells in our intestines have what we call microvilli that are like these protrusions of the cell membrane that allow for more surface area for things to diffuse into the cell.